chromosomes, and if you had 2 X chromosomes that were both active, you would get a double dose of all genes that were on that chromosome. Whereas males, we have just 1 X chromosome, so we get a single dose of chromosomes of the genes are on the X. So in the cells, in your cells that have the 2 X chromosomes, one of those chromosomes needs to go inactive. Otherwise, you get like double dose of all those genes. Okay, so that process is called X chromosome inactivation. The way that you see this all the time in textbooks is the calico cat. That turns out that one of the genes on X chromosomes actually is involved in hair color for cats. And so the calico cat, you can see like all the different, like the gold, orange, the white and the brown, like the spottedness of the cat. That is X chromosome inactivation seen, visually. And so what happens is that randomly, one of those chromosomes is going to be inactivated, and that inactivation ends up being a heterochromatin formation. So that chromosome will be tightly compacted into very tight heterochromatin. And if you saw a picture of the chromosomes in the cell, there's like this little wrinkled ball of DNA in the corner. That's the one X chromosome that's been activated. So you can imagine heterochromatin completely condensed, nothing can get to it, no activity at all, right? So highly structured nucleosome, no activity transcription coming from that at all. And then the other one is open, ready for business. Okay, so that's one example of heterochromatin to the extreme, where it's so tightly compacted and wound that it's basically useless to the cell, and that's done intentionally. Okay? So this is showing you that those progenitor cells, when you start off at the very beginning, when you're at the blastocyst stage in development, so you have like 4 cells at 8 and 16. So at that early stage is when this is happening, and so like as those cells then begin to replicate, then you have splotches of like this original cell decided on the father's X chromosome, and all of those from it got to be the orange part of the cat. And the ones over here were all decided on mom's X chromosome, and they turned out to be the white color of the fur of the cat, right? So that's the splotchiness that you're seeing on the cat, is depending on which cells decided which chromosome they inactivate, will determine what color that fur is for the cat. Okay? Questions on that? So this is an example of heterochromatin to the extreme. What you're reading in your paper is that chromatin is, you can change how tightly or loose that chromatin is, and that's showing you the dynamic nature of chromatin. Okay, so this in this case, not dynamic, inactivated and inactivated forever. Okay. All right, so talking about the dynamic nature. So I just want to talk a little bit about this. We won't go into great detail, but I think it's relevant to the paper that you're reading, so it's probably good to talk about it a little bit. So what you're seeing is that you should recognize the chromatin, the structure. So the yellow cylinder is your histone core complex, and the red is the DNA that wraps itself around twice. And the green is what are known as chromatin remodeling complexes. So these are complex, many different proteins that bind to chromatin and modify it. And so what you're seeing in the top picture is that chromatin remodeling machine is sliding the DNA around the histone complex and just moving it. Okay, so you can see that what was accessible at one point. So notice that this little blue thing goes from here to here. And what was pretty open and accessible now is a little bit more tightly wound to the complex. Okay, so if you've gotten to the end of the paper, this is the machinery that's doing what you're seeing happening in Figure 6. Right? Is that you're seeing those the chromatin being remodeled so that those CN, CS, NE, and the QLI, whatever those sequences is, those are hidden, and then all of a sudden, when they get activated, they're all of a sudden now accessible. There was some protein complex that made that happen, and these are a whole family of proteins that do this work of remodeling chromatin. Okay, so not only do they slide nucleosomes around, they can also either make more. Like you can see that this is a whole bunch of sliding to make it a lot more condensed. They can also remove the histone itself completely out of the DNA and then put it back together again. So these protein complexes do all sorts of that. They can go this way or they can go that way. So you can make it more condensed or less condensed. They do both of that. So there's a whole family of these proteins that do this work of modifying these histones. Okay? And that has implications about how much transcription can occur, right? The more condensed chromatin is, the less transcription that occurs, the more released or open or more towards euchromatin, the more transcription can occur, right? Because the more open it is, the more accessible the DNA is to polymerase and to these transcription factors that you've been reading about. The more condensed, the more hidden that DNA is. Okay, so if you want transcription to be turned off, you move towards this. If you want it to turn on, you move more towards this. Okay? All right, so let me give you one more view of that. So how does that happen? So I showed you this last time, at least with the tails. And so what you're seeing here is, I'm not asking you to memorize what these modifications are, but generally what you should get is that certain modifications are connected to heterochromatin. So when you methylate like these histone tails, that typically leads towards heterochromatin formation, or more condensed. When you acetylate specific histone tails, that ends up causing a more activated sort of version, a more released open. Same thing with the phosphorylation. And so generally what you're seeing here is that when you add negatively charged things, so this stands for phosphate, and this stands for acetyl or acetate. So these are both charged, negatively charged modifications. And we talked about that last time, is that add negative charge to a histone tail, and it helps repulse some of the DNA that's being wrapped around the histones, and that leads to a more open expression. Okay? Methylation leads to a neutral, and then this ends up being towards a more heterochromatin formation. Okay, so you can see that there is a host of modifications shown here, and we have mapped all of these. We know what they do. I'm not interested in you knowing that. I want you to realize that it's complex, and there's a lot of things going on here. But know that modifying the histones results in heterochromatin or euchromatin formation, depending on what we're talking about. Okay? Questions for me on that? Okay, so just put, you can't see it up there. Well, let me. So at the top here, so these words up here, they're talking about, so these are some of the protein chromatin remodeling complexes, the green that we were talking about. Here's two examples. This stands for histone deacetylase complex. This stands for histone acetyltransfer. So the acetyl is these guys. So these guys, this guy adds acetyl groups. This one takes them off, right? So adding acetyl groups will make it more relaxed and having transcription come on. So this is a dynamic process. If you want transcription to go, you add some acetyl groups on. If you want it to turn off, you take some off, right? So these complexes are just adding these different tags onto these tails, allowing the chromatin to get more condensed or more relaxed. Okay. So we're going to talk about transcription control at the end of this unit. And this is just the first layer. How histones are arranged on the chromosome or on the DNA is one layer of transcription control. And you're seeing that in the paper, right? Is that as transcription is being turned on or off, that's moving these histones around. And that's part of how the DNA gets turned on and off. Okay, so one more and then we'll see if there were questions or exist. All right, so what I want you to kind of big picture get is notice that this is a full chromosome at the very top. So you have your telomeres at the two ends, you have your centromere in the middle, and then you have the rest of the DNA that surrounds that. So what this is showing you is that depending on where you're at in the chromosome, that heterochromatin could be variable as far as how relaxed or tight it is. So what's always true is that at the tails, you have a pretty condensed heterochromatin, and certainly in the middle, the centromere, it's pretty dense heterochromatin. But it's variable in between there, depending on how much gene activity you're seeing at that level, at that chromosome, at that part. Okay, so these protein assembling complex that are making heterochromatin and euchromatin, they're going to be responsive to what gene activity is happening on that chromosome and where that's at. Okay? So this is showing you that there's sometimes either physical barriers, so like a centromere would be a physical barrier to heterochromatin not being able to be propagated. But typically what you see is that once you start to have a little bit of heterochromatin, that begins to expand until you get to some barrier sequence. Barrier sequence could be an example of a highly transcribed gene, something that remains open and highly transcribed. Could stop heterochromatin formation, but it can also begin to propagate or perpetuate itself to that place. Okay. All right. Questions for me on chromatin and how it's controlled back and forth with these complexes? All right. So one, yeah. Go ahead.
I guess I'm a little confused. So heterochromatin, is that bad then for us? Or is that, like, do we have heterochromatin and euchromatin?
Right, so that's a good question. So not necessarily bad. There are sections of your DNA. So remember we said our DNA, only 3% of it is actually encoded by genes. So you would expect the vast majority of our DNA to actually exist in heterochromatin, right? Is that if you don't have a use to get at the DNA, to use it to transcribe it, there's no reason for it to be out in the relaxed state. So heterochromatin exists as a place to store DNA. That's not being used. Other questions? So the vast majority of our DNA is going to exist in some form of heterochromatin. Okay. All right. So let's move away from just talk about DNA and let's talk about the process of replication. So just so you're clear about structure, right? So what we've talked about is basically the DNA itself. How is it packed? Where is it located? How do you call it? And then we're going to start talking about replication. Then we'll talk about transcription and translation and how those things are controlled. Okay, so that's where we're headed. All right, so 6. So here's goals for us. I think that, well, you can go look at them, but we'll kind of jump in. All right. I wonder if that will work. So hold on. The internet seemed to be funky, so we'll see if it works or not. But all right, so I want to show you. Okay. Marcia, you can flip a couple of the front lights for us. Thanks. Okay, so you've seen replication before. So what I want you to do is watch this and see if you can identify a couple different things. Leading lagging strands, you should be able to pick out pretty quickly. DNA polymerase, what color blob are we looking at? And what are the other components that are coming in and out of the sequence? Okay? So just watch and see which you can pick up. I think that there's. Yeah, okay. Okay, so we're going to keep this, going to spool this. So I'm going to glue it back. Turn to your neighbor, identify leading lagging strand, polymerase, and then the other blobs. Like what's the blue blob, what's the green blob, what's the things coming in and out. So see how much you can come up with, right? Go.
Well, it's just
of the polymerase. Okay, so processivity means how long does the enzyme go before it gets tired, right? So all enzymes have some binding affinity for their substrate, okay? And at some point an enzyme will say, will bind something, but it also will let it go. Okay, so the problem is that if a polymerase lets go of its substrate and gets tired and falls off, that has dire consequences. You've not replicated the entirety of the DNA and it's given up. And so the beta clamp provides additional stability so the polymerase doesn't accidentally let go of its substrate. So it makes sure that the DNA, because when you're talking about replication, you're talking about copying a very long piece of DNA, right? Tens of thousands of base pairs. So the beta clamp helps make sure that the polymerase remains attached to the strand and doesn't let go. Okay?
So if it does let go, is that how some disorders come about?
Yes.
In the genes?
Yep. And there are processes that will recognize that, saying we have a break in the DNA and we need to fix that before we go any further in replication. Okay? So in the cell cycle, you remember there's checkpoints. Those checkpoints are checking to make sure that this process has happened faithfully and to the complete end of it. Okay? All right, so when you look at this, I do this video because I think that this is the most accurate vision of what happens in replication. In the textbooks and maybe even some of my quizzes and tests, you'll see that the classic, the V, right, with leading, lagging, and top and bottom, and that there's two different polymerases going at two different rates. This is what we think is happening, and this is called sort of the factory model, where the polymerases are together in the center, and the DNA strand gets looped out. So the DNA is what's doing the moving, and the polymerase, actually the two polymerases are straight together with one another, right? You can see both of them, both the leading and the lagging strand polymerases are right next to one another, boom, boom, and the DNA gets lagged out. So what's kind of interesting with this is that if you watch the lagging strand synthesis, it's kind of crazy, right? I mean, it goes until it hits this, and then it starts back here again until it kind of hits that, right? So it gets these big loops that kind of get spooled out, and then it lets go and then restarts. So anyways, this is the most accurate vision of what we think replication happens and how that kind of goes about it. Okay.
You said those green things are called beta clamps?
Yep.
Is it called that because it's physically clamping on, in the video?
Yeah.
Okay.
Yep. So think about the, do you know what lock nut washers are? Right, it's a washer that has been cut and it's sort of twisted a little bit. Is that right? So that's what a beta clamp looks like. It looks like a lock nut washer. So there's a protein that will open up the beta clamp and then put it on and then sort of attach it. So literally just a circle that kind of makes sure that it doesn't go anywhere. Okay. All right, so that's good for that. All right, do you mind hitting lights again, Anderson? Thank you. All right. So I think that we're good on this. So will you explain the difference between conservative, semi-conservative, and dispersive? This is your Gen Bio review. Talk about those three terms to one another and the differences between them. Okay, go.
Oh, that's right, it's like right in the center. So it's the same. It's like
part of it.
Conservative? Only conservative? Does semi-conservative do an exact copy? Alright, so it says ideally replication results in an exact copy of the parent DNA to the daughter piece of DNA. So I'm just interested in which of these does that the best, or does it at all? Okay, why don't you talk to your neighbor, see what you think of. Which one? Ready?
Oh, yeah.
Bring it. Okay, let's hear it. Semi-conservative, you think? What do other people think? Both? Both semi and conservative? Not dispersive though. Okay. Any other thoughts? So far we have a vote for semi-conservative, and we have some votes for both semi-conservative and conservative, but not dispersive. Are there any other thoughts out there that you want to express? Semi-conservative, we're at Paxton. What do you think, ladies?
We said conservative.
You just said conservative. Okay. The answer is all three. They all result in an exact copy. It's just how that they do that. So if you remember, this was before we knew what was happening. These are the three different models that would result in having an exact copy, and the question of just how did it happen. You already know the answer. The answer of how it happens is the semi-conservative one, but all three of these are valid as far as this is the exact copy of that. It's just how that you rearrange the new and the old. Okay? And that's the difference between these three. That either it's all new and all old, half new, half old, or half new, half old, but mixy matchy. Okay? Good? All right. So you should be able to explain this experiment to your friend and neighbor. This is your Gen Bio review of this, and we'll dive deeper. But this, so will you? How many people this is familiar? Right? Great. Explain it to your neighbor. See what you remember. You take the first 500 pictures of this one, and that stage, you take the next 200, it's basically chop it up and then distribute it into the so certainly the chromosome will always be the same order, it's just that you'll go new, old, new, right? Like if you look down the back, as opposed to like something all new. Okay, so somehow you make a copy. So as far as how you would do it, you don't know if it doesn't happen, but like you make a copy and then you condition it, match it together. Both having the same gene, but just a mix of new and old on the same daughter. So if you feel that this question is too easy, then you should go on to the next one and draw the progression of the tubes. If the answer was not semi-conservative, but it was conservative, and if it wasn't that, it was dispersive. The data, so this experiment is the one that determined that the replication occurred through a semi-conservative mechanism, but you should be able to predict what the data would have looked like if it ended up being dispersive or if it ended up being conservative. So see if you can do that as far as recreate the data for A, B, and C if it was those other two modes. Okay? Go. You guys good? Done? Not sure? Alright, so not sure means you should either ask one another or ask me, one or the other, but see where you're at. Help? Yep.
Like, how is this end force?
Yep. Okay, so it makes sense you start off with this heavy DNA, and so that means it's nitrogen that is a little heavier than normal. So you then take those cells out and put them into new media that only has light nitrogen. The only way that when they make new DNA, the only nitrogen they have accessible to them is the N14. Yep. So because it's going to, in order to create that DNA, it needs to get that nitrogen from somewhere, because all that nitrogenous spaces that exist. Does that make sense? Yeah. Okay, don't leave.
Don't leave. So then here, they're doing the same thing, like putting it back into like.
Keep on like so the only time that the N15 is present is at the very beginning.
So then that makes sense as like, so when this would like break apart, one of these would bind with the 14, and then the other one would bind with 14.
Yep.
Have just 14, and then you have to mix it. So
Yeah.
keep this,
Yep. Yep. So yeah, color is nice, right? And so you can see it here, right? It's
Right.
double gray, and then these two dark ones are the original parental strands.
Okay.
Right. So there's the parental strand, two of them together. There's one there, and then they are here, and then you have this new population of all. Yep. Yep, that's blank. Right. Yep, yep. All right, so now I'll do that same experiment, but now don't replicate it in my conservatively, replicate it conservatively, and then throw me the data that comes up. Your brain to think. How many people need more time? Yeah. Yep. Take 2 more minutes, then we're gonna go through it together. So experiment. All right. So okay, so first class, grow cells in what's called heavy nitrogen. Heavier.
Yeah, like we're
And then so the middle one shows you that if it's semi-conservative, you expect that one change production, it rattles. So that would be
it's so strong.
It's
So I didn't even know
So in that case, you would see so what about dispersive? What would dispersive look like?
I was like,
Middle ground again. So it looks like this. So then you let them divide again. And now what you see is the next one.
Oh, yeah.
And so you like together. And that's where this light is. So that's.
and then we started passing you like, so we looked up that. But I gave Wesley a tour of Taylor.
A middle ground, right? There's now this, but now that is now distributed throughout all. You do not have two separate bands. You have this. That's the process.
I think.
This, yeah, you're looking at Cynthia. One daughter, yeah. Yeah, so they're both semi-conservative. So the semi-conservative all the way down. But what you're seeing here is like so daughter. Can you tell me where you're? So I feel pretty confident. I'm still really confused. After I'm fairly certain I can draw the answer to that up there on the board. I'm really confused. Not sure that I would even know where to start. I just need to know where you're at, so I just know how much time I process this with you. Okay. Okay. Good. All right. So were you able to? All right. So let's just walk through the example. Let's see how far you get. Okay. So on my left, let's do conservative first, and then here's A, B, and C. So up here, and then I have three bands. I have light, medium, and heavy. L, M, H. Okay. So for conservative, how do we start out? Good, all heavy, right? So we're all gonna, and we're gonna start that the same for all of them, okay? So we grow both of these in heavy nitrogen, right? So you have this DNA that is heavier than normal, and that's what that band looks like. Okay, so when you go through the first round of replication, you have now 2 copies, right? So this is just looking at 2 rounds of replication. You go from 1 to 2, right? You double, and then you go from 2 to 4, right? Because every time of replication, you produce double the amount that you have. Okay, so that's all you're seeing, 1 to 2 to 4, and then eventually to 8 to 16, all the way through. Okay, so when that first round of replication, when you're at conservative replication, what happens to this band? What do you expect as far as your low, medium, high, low, medium, heavy, or light, medium, heavy? It's going to be heavy. How many bands do I have first? Let's go with that. Okay. Pick your fingers on the air. How many bands? 1 to 5 do I have? 5. You have 5 fingers, so you got 5. 2, good. You have 2 bands in conservative. What are they?
One's going to be heavy.
One's going to be heavy, I agree.
And one light.
And 1 light. Okay? So, and that's because in conservative, if you look back here, right, you do this. You make a red one, all parent, and you make a blue one, all daughter, right? So this would be all heavy, all light, okay? So 2 bands. So with this first round of the experiment, the investigators were able to eliminate conservative as a mode of replication. They did not see 2 bands, they saw 1. So at that point, you're down to conservative, semi-conservative, or dispersive, right? As dispersive, if you look at dispersive, here, how many bands and where is it? One. Medium. Okay? Because if you're looking at the difference between this and this, as far as the amount of blue here and the amount of blue here, it's the same. It's just the combination of where that blue is at, right? This is half blue, half red. It's just that rather than one strand being completely blue, it's half of that strand is blue. Okay? So that still looks exactly the same as semi-conservative. All right, so now the next road, the next way, or the next round of replication. Conservative, what happens? L, M, H. two bands, light and heavy. All right, so are they going to be, yeah, so the light band doubles at least, right? Yeah, doubles, right? Because you have a lot more light, and then you still have this remaining one strand that is parental. Okay? Dispersive? One medium, and so this gets technical, but if these are my little marks, will the medium band be moving this way or that way on this, in the tube? Up, yeah. So it will be a medium band, but it will be slightly closer to light than it was up here. Why? Lizzie?
Because there's new stuff being made every time.
Yeah. So the only source of new nitrogen that the cell has is all light. So that dispersive, that mosaic that you see, will become more blue than red, more light than dark, or light than heavy. Okay? All right. 1 minute. Explain these results to your friends. Make sure that you understand them, and let's regather and have any last questions before I move on. Go. All right, so we're going to focus, for the most part, there will be times I go back and forth, but usually what happens, so we begin to understand stuff in bacteria, and then we go to eukaryotes and we realize that it's similar, but just a whole lot more complex. So that's normally the progression. I'll show you what happens in bacteria, because it's simplified and straightforward, and then I'll give you some indication of how much more complex it gets in eukaryotes. Okay, so here's what happens in prokaryotes in bacteria is that you'll remember that bacteria have one circular chromosome, and replication proceeds from one set point around bidirectionally until it gets down to the other end of the circle, and then the two circles are then decanted. They actually end up being together like this, and they separate them, and then you have two copies of the chromosome. Okay, so because the reason why we know so much of this is that there's a lot of mutations that we have found that kind of stop the process along the ways, and that has kind of illustrated what actually the process ends up happening and what happens in that process. Okay, so we're going to look at this. The replication occurs. It starts at what's called an ORI, or an origin of replication. There's a sequence that all replication begins at, and it's similar in all these bacteria. And then the replication fork splits and goes bidirectionally around the circle. Okay, so we're going to walk through how that occurs. Okay, so here's another view of that. Here's the origin of replication, and here's the bidirectional fork. So you can see that there's a replication fork here and a replication fork here, right? So one of these will be leading and one will be lagging, and so you'll have to do this leading lagging scenario. And then once you get to the end, the process is just letting both of those daughter chromosomes go to two different cells that they're part of. Okay. All right. So so let me talk. So that's the general process. Let me talk about the players that actually do that process. Okay. So you're beginning to understand some of this. So what you'll know in prokaryotes is there are various forms of polymerases. Okay, so you've been reading about Pol II, talking about transcription, starting with DNA, but these are different polymerases that you can see what they end up doing. So Pol I is responsible for doing the primer, like setting down the actual RNA primer or the DNA primer before that starts. Pol II is involved in the damage repair, and Pol III is what does DNA replication. So all these enzymes do the work of adding down DNA bases. They're they're specialized in the jobs that they do. Some are only doing replication, some are only doing damage repair, and some are only doing these, setting down these primers. So generally the same enzyme doing different work. Okay. The other one, so you can see that all the other players over here, DNA, helicase, primase, all these sort of stuff, these are all present in both eukaryotes and prokaryotes. It's just that they're more complicated versions that exist in eukaryotes. Okay. All right. So notice that in order for this to work, you have to have a DNA template to actually begin, and it always occurs 5' to 3'. That should be something that you probably remember, is that replication always occurs in that 5' to 3' direction. Okay, which is why that you have that lagging strand synthesis. Okay, so notice that so when you have this, what I'd like you to think about is, here are various forms of templates of DNA. I want you to tell me which of these could serve as a template for DNA polymerase. Okay, so here's your DNA polymerase, what we talked about, right? What they require is that they have to assume that they have a DNA strand, assume that they have magnesium chloride, all that sort of stuff. Which of these would actually be able to prime DNA synthesis? Okay, does that make sense? So you're looking to see which of these would be good that we could make a copy from. Okay. So another kind of sub-question under that, for those that wouldn't do DNA replication, come back here, you say, we've looked at these 3 different polymerases. Which of these polymerases could act on these templates? So see if that question kind of has some thoughts for you as well. Okay? We'll give you a couple minutes to think about that, talk about it with one another, and then we'll come back. You're going to start here and move this way, so that we're going to add a DNA here, 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 all the way around. So a free 3' end like this, that's what you're looking for. There's no free 3' end here to add anything to. Okay? What about this thing? No. You could fix that if you want, but you're not going to do replication from this place, right? Because you need to unwind this and get rid of it. What about this guy? Yeah, I mean, it would be a short run, but you could do it right here, right? This is the free 3' end, so you could replicate the last parts here. This one?
Yes.
Yes? This one? No. Okay? All right, so what you're looking for, right? You need a free 3' end in order to add to it, and then that DNA polymerase can take off from that place and move forward. Okay? All right, we'll stop there. Have a great weekend. Don't read too many papers.